Hello, everyone, and welcome back to Lady Scientist Podcast. I'm your host, Jocelyn Pearl, and today we have a great guest in store for you all. I speak with Dr. Madeleine Oudin. She's an assistant professor of bioengineering at Tufts University, and she takes us through her career journey from undergrad at McGill to getting a master's and going on to get her PhD in neuroscience. She then shifted gears and studied cancer in her postdoc at MIT, and then went down the very challenging and competitive path of pursuing a faculty position. And she is, of course, successful in that journey and has been mentoring others and leading science in this incredible niche environment of cancer and neuroscience, really merging those two fields that she worked on. She also became a mom at some point during that busy career, and her daughter was diagnosed with a rare form of epilepsy caused by a mutation in the SCN8 gene. So we talk about her discovery of her daughter's diagnosis and how that's shifted her role as a scientist and patient advocate, and what some of the resources have been for her and her husband as they go down this path of drug development for their daughter. It's a really inspiring conversation, and I hope that you take something away from Madeleine's experience and her journey to becoming a professor. Thanks so much for listening to Lady Scientist Podcast. Today, it is my pleasure to welcome Dr. Madeleine Oudin. She's an assistant professor in biomedical engineering at Tufts University, and I'm excited to talk about her career journey and her life as a parent today. So welcome to Lady Scientist Podcast.
It's great to be here. Thank you for having me.
So I want to start out at the beginning, the very beginning for you as a scientist. What led you to pursue a career in science?
I always liked the science fields. My dad was really into math, so I did lots of math growing up, and but then I really got interested in biology and chemistry and medicine and how the body worked. So I was watching ER at the time and loved that show and the medical profession and just kind of trying to help bodies and people get better and the science behind that. And so in high school, I pursued science more actively and just really enjoyed biology and chemistry, and thought I wanted to go into medicine at first. And so, but at least as my undergrad, focused really on that.
So you had already kind of a supportive environment in high school. Did you have great science teachers, or what kind of school did you go to?
So I grew up in Europe, in France. My mom is Canadian, my dad is French, and so I went to an international school. So I did have good science teachers. I mean, my family at home was not into science at all. I had no exposure to science careers or anything like that. So it was really my own passion and interest in medicine and science. But we did have a very strong science curriculum, and in France, you have to decide between either the more literary track, more economics track, or more science track when you get into high school. And so I picked that. And so we had intense, biology, chemistry, physics, math. And so we learned a lot. We did a lot of labs. And so in that sense, I got a lot of exposure to that world. But that's really it. I didn't really know what kind of careers. I thought medicine was the only career that you could do. I didn't really know that you could become a scientist. So I didn't really have anyone to look up to or anyone to kind of, see which way to go. It was really just for my own passion.
Did that change when you were in your undergraduate studies? And where did you end up going for undergrad?
So I ended up going to Canada, to McGill University. I wanted to explore my Canadian heritage. And so I went there, and McGill was great, but it's a huge school. So my classes were humongous. The smallest class I had as a senior was 60 people. And we always had rotating professors come in giving all these guest lectures. So while I learned a lot, I still then didn't really get a lot of research exposure. We didn't really have advisors like I advise undergrads now. I really didn't know, again, that you could start working in a lab. Like I was clueless. I had no idea. I had a passion for flying trapeze and did that during the summers during my undergrad, teaching flying trapeze. I just didn't know you could pursue research. But as a senior, or in undergrad, I started to learn more in my classes, reading papers, learning more kind of about the lab. And so while I thought I had been wanting to go to medical school, and kind of was pursuing that more, I realized that the idea of discovery, understanding how things work, that was really exciting to me. And so I wanted to start doing research. And so by that point, it was the end of undergrad. And so then I decided to do a master's to get research experience, because I was told that if I wanted to do PhDs, I needed to have more experience. And I had none. I really had done no research apart from my classes. And so I did that. So I went to do a master's to gain some research experience. And that's really where I fell in love with research and got that exposure finally to really what it is to be a scientist. So it took me a while to get there, and I really didn't have anyone showing me the way. So yeah, it took me a while.
Jumping ahead a little bit, I'm so curious. I mean, because obviously you're a PI now and a mentor to many, many people, I imagine. What advice do you give people going through that transition themselves?
Yeah, the number of students that come in wanting to go to medical school as a freshman because they think that that's really, the main career that comes out of liking science, to the ones that then as seniors actually going to medicine, it's been interesting with my first crop of advisees. Initially, 90% wanted to go to med school, and now it's probably like 10%. So a lot of them have just discovered all the other careers that exist along the way. So I really encourage my advisees to get involved in research and to try working in labs or do internships, especially over the summer. It's good to get involved during the semester, but obviously they're busy with classes. So to really taking advantage of those summers to try different types of research, working in academia, working in industry, and trying different fields, just to get that exposure to really figure out what you don't like and what you do really like. And I think that's really what helps the most to, figure out what you want to do next.
So you had this master's under your belt, you had gotten some initial experience in the lab, you knew this was really what you were passionate about. You go on to get a PhD from King's College of London studying neuroscience. Can you talk about how you made the decision as far as program, where to study? You've already had this international experience. Like I'm so curious, like how that all played into your decision there.
Yeah. So I ended up going back to the UK for my master's, and then stayed for my PhD. So when I was looking for master's programs, at that point I was kind of missing Europe, having grown up there and been in Canada for my undergrad. That was fun, but I thought, it would be cool to go back to Europe. So that was more of a personal decision, just to, move and try something different. And so I did a master's there, and then I ended up doing a PhD in the same lab that I did my master's research project in. So I got to work there, and the advisor, we got along really well, and he saw that I was really enthusiastic and really motivated, and so he offered me a spot. And in the UK, it's changing a bit, but it's more usually direct admission into a lab. So I kind of knew I was going to that lab and into that program, and so stayed to do my PhD there, studying neuroscience and the role of endocannabinoid signaling in adult neurogenesis.
For folks who aren't familiar with, program selection, lab selection, can you explain a little bit about the differences as far as going into a program versus joining a specific lab, and what that looks like for people who are trying to do a PhD?
Yeah, so you can either join a PhD program where the first year you might do rotations, and so work in 3 or 4 different labs for 4 to 6 weeks, depending on the program. So you kind of get exposure to different labs, and at the end of their first year, you decide which lab you want to join and commit to. And so the other way to do that is that you actually apply to a school, but work with a specific PI in a specific lab. So you are part of a larger program, but coming in who you're going to start working with, what projects you're going to be working on. And so sometimes it can be field specific, like in biomedical engineering where I am. there's less rotations and more direct admissions, but there's still some. But in biology, it can be much more rotation based. So there's pros and cons to both systems. If you really know what you want to do, it's nice to be able to just get started and not do the rotations. But if you're not quite sure, and then you can get exposure to different labs that you might not usually think that you would be interested in, and that can maybe change your mind too. So there's definitely advantages to both ways of doing it.
Yeah, and I think that's often one of the things that can be a little bit confusing for students coming in. And for my mentees, I like to remind them for particular programs in the States that they will have that time their first year usually to do rotations and to work with a few different PIs and start to figure out what makes sense for them, because it is a big commitment to then join a lab and be working for this person for several years and be focused on that particular area of study. So I think
Yeah,
it's great to
absolutely.
share, those different experiences.
Mhm.
So tell us a little bit about what your work was focused on in graduate school.
So I was studying adult neurogenesis. So for a long time, we thought that once you were born, you had all the neurons that would be in your brain. But it turns out that even into adulthood, there are a few regions in the adult brain that continue to proliferate and generate new neurons. And so we were studying this process and what these, how do we get more of these neurons? What do they do? Where do they go? And we were studying the role of the endocannabinoid system. So these are the receptors and proteins that cannabis acts on, but we have our own molecules within our bodies that function and can activate the same receptors and are very important for maintaining brain function. And so we were studying the role of that pathway in this process and trying to stimulate this production of neurons, because the idea was that, if we could generate new neurons in the adult brain, they could repair ones that may have died due to an injury or stroke. And so that could obviously be attractive for various regenerative approaches.
I imagine from a biological perspective, it's a bit of a delicate dance, though, too, because if there's too much neurogenesis, that could maybe lead to cancer. Am I saying that correctly?
Yes, definitely. Yeah, it's always a balance for sure.
Interesting. And so you finish your PhD and you go on to doing a postdoc in Frank Gertler's lab at MIT. Walk us through the decision to move to studying cancer and going to MIT now in Boston. You're really like traveling the globe as far as your
Yes.
research.
Yeah, so I really, liked studying the brain, and we were studying how these cells, once they're generated, move to different regions of the brain. And so I got to learn how to do live imaging with microscopes, where we would watch cells for several hours and see in which direction they moved. Could we get them to move to the places that we would want to? So I really enjoyed that. But these regions of the brain that generate new cells, they're very small. We still don't really know what impact they have. And the one region I was working on was very prominent in mice, because it is linked to a sense of smell, which the mice have a very strong sense of smell. But as humans, we don't really have a strong sense of smell anymore. And so that region, we didn't really know what it did in the human brain. And so I just didn't feel like it was impactful enough. I didn't feel like what I was doing, could benefit people. I want to do something that had more of a direct clinical impact. And so when I was thinking of postdocs, I was looking around and I attended local meetings on called the Cell Motility Club, which are meetings of researchers that work on cell motility, so how cells move in different contexts. And I learned that cancer cells move when they're spreading, when they metastasize. And that's really a process that we don't understand well, that is, very, that leads to patients dying. And so I decided I wanted to apply my knowledge and expertise of how neuronal cells move to cancer cells. So I started looking for labs that, studied cancer cell migration and that also used a lot of imaging and microscopy live inside of tumors. That was the method that I wanted to learn how to do. So it was both based off my interest and also of the topic, but also the methods that I wanted to learn. So there are a
Wow.
few labs that jumped out, and then I applied to those, and I ended up joining Frank Gertler's lab at MIT.
Can you give us a picture of I've never done live tumor cell imaging before, so if you could just share what that is like. What's the kind of broad strokes protocol, and what type of information are you able to capture?
Yeah, so we can basically have fluorescently labeled tumor cells, and that so we generate tumors in different tissues. So we worked in breast cancer, so in the mammary glands, and you can anesthetize the mice once they have these tumors, and you can expose the tumors by doing surgeries to remove the top of the skin, and then put the tumor on a 2-photon microscope. And you can keep the mice alive under anesthesia, and then you image the fluorescently labeled tumor cells in the tumor, and you can see them moving and leaving the tumor. And so we can do different treatments either to the mouse or locally in the environment, and see if that makes them move more or less. And if you have more movements, you assume that more cells are leaving the tumor and metastasizing. So that's kind of what we want to stop. So yeah, that was a technique I was really interested in learning, and not that many people do. And so I loved being able to see the cells moving, see the biology live in front of me, and that was really fun.
That is so interesting. I mean, I'm really curious. You talked about stopping the metastases, stopping these cells from mobilizing and going to other parts of the body, which of course is this like crucial leap in cancer that causes, you know, the more extreme pathogenesis of the disease. Are there particular gene targets that you've found that you can then target drugs to potentially stop metastasis?
So yeah, it's tricky because it's not a single mutation or gene that's associated with metastasis. So it's often a combination of changes in the environment that happen when there's a tumor that can then attract tumor cells more, and also these cells, expressing several genes or activating several pathways that can make them more responsive to these outside cues and more able to survive in these foreign environments. So there's definitely, some new drugs that are being tested and emerging, targeting those specific pathways that are more associated with motility, or trying to alter the local environment in such a way that, it would be less attractive for the tumor cells. But yeah, it's definitely challenging.
Interesting. Really interesting work. So, of course, you're now a PI at a university, and this is a, highly competitive landscape to go out and get these types of positions. What was it during your postdoc that gave you the conviction that you did want to look for a faculty position? And were there particular pieces you had in place, grants or mentorship that you felt like I'm ready to do this?
I took my time as a postdoc. So in the end, I stayed 6 years. I didn't apply till my fifth year. And so I really took my time to get comfortable in the cancer field, to publish papers. I collaborated with a lot of people in my postdoc. So being at MIT, I got to work with a lot of engineers, which was not something I'd ever thought of doing, but they're there, and I really took advantage of that. And so I got to apply lots of different engineering methods, like computational approaches, devices, to studying cancer biology. And so I really developed kind of a different way of studying cancer, leveraging these different approaches. And the environment at the Koch Institute where I was fantastic for that. And so, I took my time, and I really learned a lot and developed, a broad skill set and, papers to back that up. And so along the way, I definitely continued, felt that academia was an option. I wasn't 100% committed. I wanted to see how things went. I was fortunate to apply for funding and receive initially a DoD breast cancer fellowship when I first started, which funded my work, and that was really gratifying to get funding. I applied to 8 fellowships, and it was the last one I got, so it was not easy, but I persisted, and I got that. And then along the way, I also mentored students and tried to see if that was something that I really enjoyed. And I really did like that aspect too. So I tried to kind of gain practice in these different areas that I knew would be important to be a PI, and see if I liked it. And then I ended up applying for the K-99 grant, which is the Pathway to Independence. And so that I, put together what I would want my lab to work on. I think sometimes there's a lot of trends going on in research, and you think that you have to, follow what everyone is doing. And that's not what I wanted to do. If I was going to start my lab, I wanted it to be on metastasis, on the tumor microenvironment, on the extracellular matrix, these things that I was really interested, which, are, it's not that they're not popular, that people think they're important, but they're not, the trendiest thing at the time. And so, but I was like, I'm going to start my lab. I want it to be on something I'm passionate about. And so I wrote my research proposal, and I took the time to write it. I really put in a lot of effort to think about what I would want to do. And I submitted that, and I waited. And in the meantime, I actually got a job offer from a local biotech from a faculty that I knew who was starting a company and wanted me to lead the biology. And I was really tempted because I really looked up to her as a mentor, and, thought this could be super fun too. And I was pretty much ready to go there because I thought my grant would not get funded. And, and then I got the score for my grant, and it was really good, and I knew it would get funded. And so I felt like, well, if the NCI, is funding me and believes in me, then I should believe in myself too. And so I decided to turn down the job in biotech and to go out on the job market. And that really gave me the confidence, in addition to my publications and the experience that I had, that, I had a good skill set to start in academia and to apply for jobs.
Wow. Incredible. It sounds like, it was really 50-50 for a while there as far as industry or academia. It's really interesting to hear that. Um, can you talk about what advice your PI was giving you, and maybe even your PhD advisor, and just how that played into your thinking?
Yeah, I had great discussions with both of them. Balancing, whether to apply for positions in medical schools where, you don't teach, you have, this different salary structure where you have to come up with more salary from grants, versus being in a department where you do teach and, you get more salary support. And, those discussions like that, because when you work, in biology, you can kind of end up in lots of different places. Also, I really enjoyed engineering. And so I thought it could be fun to be in a biomedical engineering department as well. So I was applying to those. That was a bit harder because, I'm not a trained engineer. And so some departments were just not really interested. And so kind of navigating which one was a good fit. And, just, applying to different places. Everyone always says you find the right fit, and that can be a little hard to describe, but really kind of taking advantage of the interviews and talking to people and just, seeing what kind of questions they ask you. Do you really feel like a good scientific vibe, in that environment? And so, because I did kind of apply a bit later, and I had published papers, I felt like I was really able to take the time when I was on the job market to, research and be really ready when I went to places and have a lot of questions, and just take the time to really think about where would be the right place for me and my research interests. And so my mentor really supported me in that, and was always available to chat and help me to make that decision.
What was the interview process like for you? Were you giving chalk talks, like maybe just a few highlights of what the process is like for anybody who is thinking about pursuing this type of track?
Yeah, there's, intense applications up until usually, October, November, depending on some places. And so, that's really busy just submitting and, tailoring a bit each application for each school. And then there's a bit of a wait period, and then interviews start. And usually they'll start in January, but mine actually started in December. So, and some places will have one interview where they put in your seminar and a chalk talk where you present your research plans in the same day, and some do it over 2 visits. So it depends. Mine were mostly 2 visits. So the first visit was just a seminar, meeting people, and then got invited back for a second visit where I did the chalk talk, and that was in the new year. And, met more faculty, started talking a bit more logistics, equipment, what do I need, what space would be available. So, because there's also a lot of that aspect to it, which is, what do you need for your lab to be successful? Do they have the right core facilities and equipment to support you? And so there's also kind of that very logistical aspect that is important. And so you kind of have to weigh all those factors. Obviously, there's location, where you want to live, some places where they're expensive versus salary. Then, there's the community, there's the resources. So there's just a lot of factors that go into it, into, making that decision. So it's a very long process, although mine ended in March. So it was a little earlier. Like I said, all the places I ended up interviewing at were on top of everything and starting interviews very quickly. Sometimes it can keep going till April, May. So it's a long process. It's very busy and lots of traveling. And so I was lucky. It was pre-pandemic and I got to go everywhere, which I loved, because some places I had never been. So that was important to see if I wanted to live there or not. And so, I think it's important to really take the time, because it's a huge decision. You're committing to being there potentially for your entire, career. And so you want to make sure that you spend the time and make a good decision for yourself.
Yeah. Wow. Well, it's so impressive. Your career path, and hopefully your insights will help inspire others and make them feel like they can put the pieces together and go down that path if that's what they're interested in. So obviously becoming a PI is a lot. I think from some of the other folks I've spoken to, often those first few years of running your own lab can be very challenging, isolating sometimes, as you start to form this new community at a new university, and figuring out, what it's going to take to run a successful lab. I'd love for you to actually just share a few highlights for yourself over these last few years, and either research or mentoring highlights to help inspire others.
Yeah, it's a whole kind of new journey, and you go from being a postdoc running experiments to the next day just running everything, and you're the boss, and so I think definitely, hiring the first people in my lab, you're like, first you're like, oh my gosh, you want to work with me? This is so exciting. And, getting everything set up to run some of the first key experiments in your lab. Those are just so exciting moments. And, research wise, I think having your first lab paper where you're the senior author, that's all research from your lab, it's just so exciting. And putting that out there and, seeing people actually read it. So we published our first two papers in 2020, and they've been downloaded almost 4,000 times each, which is crazy, and it's so exciting and validating. And I think having people comment positively after you put in all this time, and these are your ideas that you're putting out there, that's just really exciting. I think, getting grants is obviously a huge part of becoming a faculty, and it's very stressful because there's so many rejections. And I still remember the first grant that we got, and it was early in the morning. We got the email, and no one was at work. I was in the hallway. I'm like, I need to tell someone, and calling my husband. He's not responding. I'm in my lab. No one's there. I'm like, I just need to yell this that we got a grant. And that's also just really, really exciting to have that validation, that, with your ideas that you can get funding. Because, we've been lucky, but I've written a lot of grants, gotten a lot of rejections that are really, really hurt, really things, attacking your science. And so, that's just really hard. So you really have to celebrate all the wins. And I think that's something that I really do a lot with the members of my lab. We always celebrate, passing PhD quals, birthdays, obviously papers and grants. And I think that's so important because there are lots of tough times and rejections in science. And so learning to deal with that is hard, and celebrating all the wins. I've been lucky. I've had some amazing people in the lab. This past year I had 3 PhD students graduate, which, is awesome to see them, to have had them in the lab. And when they just started and grow to be, mature, independent scientists and move on to their different careers, has been really, really exciting for me to see that. And so, all these people that have been part of this story of the lab, are all important to me, and I love watching them, going on in their careers.
Wow, that's awesome. I want to shift the conversation a little bit. Somewhere along this very industrious path, very exciting career, you became a mom. And just for some personal backstory, I actually gave birth to my daughter 6 months ago. So she's 6 months old, and I've been going through this transition myself of going from, having a life that's focused on my work and my passion to now having this wonderful daughter in my life and getting to learn about being a mom and having that be a big part of my life too. So I'd love to hear how, having your daughter changed your relationship with your career and kind of your life.
Yeah, so I was pregnant during the pandemic, so it was kind of a weird time to be pregnant because no one saw me at work or pregnant, or we were all working from home, so it didn't seem as real in the work world, because we were all working from home. But my daughter was born in March 2021, and it's definitely, when you're on maternity leave and you're running your lab, like you can't really disconnect completely because you're the one running your lab, and I wanted to, but we had papers to submit, we had things going on, we had been already delayed with a pandemic. So I pretty quickly had to learn how to balance being a mom and taking the time to do that, but also balance having the lab and running a lab, because again, the lab doesn't stop. And I took the time that I needed, but I also was excited to do some science too, because when you switch from being like a full-time scientist to full-time mom, it's really weird because it's not what you're used to, and you kind of so it was nice also here and there to be able to stay involved with my lab during my maternity leave, to not completely lose kind of the life that I had. But and so initially, it was pretty good. She had an easy delivery, and she was a chill baby, so life was pretty smooth. But then at 3 months old, she started having seizures, and they were little seizures, but we noticed them and thought they looked weird, and we took a video and sent it to our pediatrician, and she told us to go to the emergency room at Boston Children's. And so we went there, and she was diagnosed with epilepsy. But we got the seizures under control, so we didn't think, we were like, oh, it's just a fluke. We were trying to find a reason for why this had happened. We'd actually just started a reflux medicine, and we were looking up, and there were some papers suggesting that it could, like some obscure papers, like we were trying to find a source of reason. And so, we were okay. But when we were at Boston Children's, they said, we can send for genetic testing for seizures. And at the time I was like, okay, there's nothing wrong with my child. Like, but if you want to go, and do that, sure, why not? Like, it's always good to get information. So they sent it out, and 5 weeks later, they called us and said, your daughter has 2 mutations in this SCN88 sodium channel gene. And then, our whole lives changed. And so, what initially was kind of very smooth sailing motherhood, like things are going really well. I was, really felt like I was managing. Like I said, I had this great maternity leave. I was still able to do some work. She was a good sleeper. Life was good. I was managing this whole mom baby life. And then everything changed.
Wow. Wow. Um, so I'm curious what happened next, because I think many parents who are going through this experience of having a child with a rare illness or genetic mutation do not have the scientific background that you have. And I know that your husband is also a researcher as well. So I'm curious what, what were the next steps for you? Like, what are we going to do about this? How are we going to move forward with looking for therapeutic options or, giving her a, high quality life?
Yeah. So as soon as we got the diagnosis, thankfully, actually, the fellow, normally they wait for a genetic counselor to tell you, but we were hounding him, and he knew we were scientists, so he gave us the mutations, and then of course we went to PubMed, and we started looking up papers, the gene, where the mutations were, and initially it was a grim, it was a really tough day because mutations in this gene, there's about 800 in the world. There's a range of phenotypes, associated with mutations in this gene, but it can go from, very severe seizures, and then also very halted development. So it's not just the seizures, but it's also, not developing in terms of, having any muscle control, walking, talking, communicating, all these things. And so again, there's a huge spectrum of kind of these different mutations and what phenotypes the children end up having and how they evolve. And so, it's a huge range. Now, the thing that was, really that we were lucky was that this gene has actually been studied for a long time. So it was discovered in the 1990s by a researcher in Michigan, and she had found it in mice and had was hypothesized that it could be associated with epilepsy activity because it was such an important regulator of neuronal activity. And then it wasn't until 2012 when actually another scientist parent, Michael Hammer, his daughter had seizures unexplained, and he did whole genome on his entire family and himself, and found that his daughter had a mutation in the SCN8A gene. And he, connected with this researcher in Michigan, and they started working together. And so, we were lucky that our gene, has such a rich history of scientists working on it. And there's mouse models and antibodies and a lot of resources and a patient registry. And so, right away, we were able to read a lot of papers. We were able to connect, with all these researchers who really welcomed us in and, shared lots of information, predictions on what her mutation would do, connecting us to people who could test the function of her mutation so that we could really understand the biology behind, her sodium channel dysfunction. And so between that community and then our expertise, because, it turns out that I had started working on the role of ion channels in cancer several years before and interested in how the electrical properties of tumor cells can shift. And so I had done work on potassium channels, mainly a little bit sodium channels. And so I was kind of, familiar a little bit with this space. I was on my PhD in neuroscience, so this wasn't completely foreign. And then lastly, after some digging, my husband found that her mutation was in this alternatively spliced exon. So this region of the RNA of which there are different versions of the protein. And he, figured that out right away. And there's a lot of interesting biology associated with that. And he's been working on splicing for 20 years. So it was just like between the knowledge of the community, like my expertise, my husband's expertise, and then lastly, the fact that Tufts, where I am, has a huge epilepsy community with researchers who are very strong in the neuroscience department that I was able to connect with right away. We like sucked all this knowledge in and have been just learning and could really kind of within a few months really get a handle on, where we were at, what we needed to know, what we needed to do, and kind of get to figure out what options were going. So, it's definitely a surreal experience to have this much, this coincidence of like where her mutation was and the expertise that we have. And, and I'm a scientist. I don't believe in, I'm very fact-based, but I feel like there's some kind of fate in this in some way. And that, we were meant to come and then contribute to this field.
Wow.
So yeah, I think we experienced this really from the science side, and it was very, very, very different. But yeah.
Well, I find it so heartwarming too, just, the access you had to, a clinician who was willing to do the genetic sequencing right away and give you the information. Like, I feel like that's also kind of a rare occurrence for a lot of patients who often have to kind of uh, battle to get that information, to argue to their insurance companies, um, and are often waiting a long time for that ultimate genetic diagnosis for symptoms that they've been aware of for quite some time. So, um, and it's so, I think, also wonderful that you had this community that you stepped into that already had all of this, expertise and experience with the Hammer family. And like, one of the things I find really inspiring about patient communities is how much research has been done for a lot of these illnesses, and like how that's really driven forward entire fields. Like, if you look at Huntington's disease, for instance, that was like how molecular biology became connected to clinical diagnosis, and founded, this kind of entire space in a way. So I want to fast forward because you posted recently that you traveled to a conference, AES, and presented your very first epilepsy talk. So obviously, with this information in hand, you have now started studying epilepsy. Can you talk about that shift? What aspects of the biology you're trying to understand further with your research, and maybe just a few highlights from the conference?
Yeah, no, I really pretty quickly decided that I maybe did want to contribute. And I was also inspired on Rare Disease Day in February this year. There was a post from another academic at Penn who talked about his daughter and rare disease. And so then I looked him up, and he had also started as an assistant professor research on his daughter's disease. And I was able to meet with him a few weeks later. And, he kind of showed me and convinced me that I could do this, that I, could start this new area in my lab, because, as an assistant professor, you're still establishing yourself. And, my lab was doing well at that time. We published papers, we got grants. So I felt like I was on solid ground, at least in that sense, to get started. But it was intimidating. And so between that and then my collaborator at Tufts, I was able to, really want to start doing that. So I was able to recruit a PhD student who started working in my lab in June on this project. And so we're studying the role of the splicing regulation of SCN8A. So there are two versions of SCN8A, this gene, an adult version and a neonatal version, and it switches over time. So you have a neonatal version that's more abundant at first, and then it goes to an adult version. And so my daughter's mutation is in this part that governs the neonatal version, but there's about 35 other patients that have mutations in this region as well. And so we are, trying to switch to the adult version to remove the mutation completely. And so, there's a lot we don't understand about how this switch happens, how is it regulated? What does it do? So we're doing lots of work on that. And, my husband is collaborating and an advisor as well. So it's really a family project. And then we've also started generating a mouse model with Margot's mutation. So with a mutation in this neonatal version, so that we can study the effect that has. And if we switch it in the mouse, what happens? So that mouse will be ready in a few months. And the technology we're using are ASOs, antisense oligonucleotides, which have become a really popular and really cool tool to manipulate gene expression. And this has been pioneered really by the company Ionis. And so thankfully, because my husband works in the RNA world, we know lots of people in that community as well who've given us a lot of advice. And we've had so many friends of ours, scientists, who have stepped up to do experiments in their own labs too, to help us, like generating iPS cells for Margot's fibroblasts to study make neurons, things like that. And so, it's been really amazing to have, all these other scientists get involved in this research as well, that were not doing this before and getting involved in SCN8A research. So we are working on that. And we also applied for the Enlorum Foundation for Margot, which is a foundation that kind of spun out of Ionis to generate N of 1 ASO treatments for genetic disorders for nano rare patients. So diseases where there's only, that 1 or less than 10 patients with that mutation. And so Margot was accepted into that. So they're also generating ASOs to test on the human side. So in my lab, we're kind of more focused on the mouse side and understanding the biology and the functional effects of these changes. And then they're developing treatments there. And so our clinician is involved in that. And he's been awesome too. From our first meeting, when I knew we were meeting with him after her diagnosis, I had already looked up a bunch of his papers and came to our first meeting. And he was like, yeah, we recently did a study. I'm like, yeah, I read that. So he, but thankfully, I think he appreciates our enthusiasm and that we're doing this. So he's the lead, PI on the human side for Enlorum. And then my husband and I are, doing the experiments. And so at the annual American Epilepsy Society meeting, they had, there's many genes have kind of their own gatherings related to epilepsy. And so there's an SCN8A gathering with families and clinicians and researchers and biotech companies. It was so awesome to have kind of every stakeholder within this community together. And we got invited to present our work. And so it was our clinician and my husband and me, and it was super. So it was very preliminary because obviously we just started, but just kind of showing what we were doing. And that was just really exciting and a big kind of accomplishment to be in this space and to have started this research and, be moving forward. And we got to meet a bunch of other families that have been, dealing with the same disorder, meeting all the researchers. So I'm just, it's really exciting to be so involved. It helps me feel more in control of, the situation when I can't control my daughter's seizures or diagnosis or anything. And so here I'm actually, contributing in the way that I can. So it's been really rewarding in that way. And I bring in also my cancer expertise, because there's a lot of similarities between cancers and neurological disorders, and that's kind of something that I've been interested in for a while, given my background. And so I'm really kind of delving into that. And drugs that are being used for cancers are being tested out for certain neurological disorders and vice versa. Mutations in pathways that are found in cancers can also be found in epilepsy disorders. So there's actually, a lot more similarity than I expected. And so I'm kind of delving into that and taking advantage of my expertise and kind of carving out this new area for my lab and for my research. And it's been really exciting.
Wow, that's awesome. On the research side, I know you recently published a review about this niche, this overlap between cancer and neuroscience. Can you highlight a few parts of that review for us, for people who are not familiar with this like super exciting new area? Because I just think, again, for my background, I studied neuroscience from a systems biology perspective in graduate school, and looking at particular gene expression pathways that were changing in things like Huntington's disease or psychiatric illness. And that was something that came up a lot, where a lot of these hits were, also related to cancer. And there were folks at our institute on the TCGA project studying, these cancer pathways. So it's like when you start to think about it from a translational perspective, how do we go about targeting these things or creating better therapeutics that, create this like balanced cellular environment, which I find really interesting. So I'd love to get your highlights of this new field.
Yeah, I think it's so interesting. We know that in tumors, the local environment really drives progression. So the immune cells, the local resident cells, the extracellular matrix that provides support to our tissues. But another component that's there are nerves. And so obviously we have nerves everywhere in our body. And it wasn't just until about 10 years ago that there was one study showing that these nerves in these tissues, initially in prostate cancer, can drive cancer progression as well. And so now in several peripheral organs, we know there are nerves present, but when tumors form, sometimes you have more of the same nerves, sometimes you have different nerves coming in. So there's more nerves when there's a tumor. So why does that happen? Why do the tumor cells make the nerves come in? And we found that, when there's more nerves, the tumors tend to be more aggressive. And so how does that happen? How do the nerves actually impact the tumor cells? So that's, a huge area. And then there's been work that's been done in the brain, obviously with cancers that form in the brain and interacting with the brain cells, but in the rest of the body as well, we have nerves. And so this is still a really new area. So we wrote a really nice review on kind of what we know, what are the different ways to study this, because merging cancer expertise and neuroscience expertise to build models with both of these, it can be quite challenging because people come from different sides. And so we were kind of summarizing where we're at in the field with that. And then, there's also just, so there's the nerves that are present, and then there's the properties of tumor cells that make them nerve-like, like the genes they express or their electrical properties. And so, I think because there's lots of drugs out there that target neurological processes, potentially those could be, repurposed for cancer in different ways. And so, and maybe even, vice versa. So one of the drugs that my daughter's on, a sodium channel blocker, I was looking up papers on it, and it's been shown in a mouse model to breast cancer metastasis. So that's, kind of interesting to see these. So I think, there's a lot more that we can learn, from looking at both of these different areas. So different way of looking at things to learn more.
Yeah, that's so interesting. I think it's so neat that, your graduate school and postdoc work kind of led to you having this expertise in these two different areas, and now, really drilling into that and defining some of these new cellular models to study these things in the lab. It's really cool. I want to get back to the drug development for SCN8. Um, ASOs hold so much promise, I think, for treating a number of diseases. And I think I'm particularly passionate and excited about them for neurologic disease. But of course, there have been some challenges there too. We've seen, a couple phase 3 trials that were halted for Huntington's disease and I believe Rett syndrome. And, I think one of the questions is like, will we be able to get these reagents into the brain? Will we have the coverage we need to see an effect? And, will we do no harm? Which there was, of course, a recent, trial with 2 patients with epilepsy that had some serious adverse events. So I'm curious where your head is at with this field, and um, as a mother and patient advocate, like, how hopeful are you? Like, how close are you to having a type of reagent that you think would be worthwhile to try?
Yeah, I mean, I think it's still, I'm not an expert in the ASO field, but coming in from the outside and as a patient, I think there's still a lot we need to learn. I think with Enlorem, we trust this is a company, they're working with Ionis that has been working on this for many, many years. So they have lots of experience. They were able to bring, Spinraza for SMA, to patients, and that's been working really well. For, the N of 1, it's obviously different, and it's, getting into the central nervous system, like you said, with all the caveats to get, enough effects. And so they have started injecting their first patients in the fall. And so I think it'll be, really interesting to see, what they learn from that, and, seeing if they can, have more effects. I think there's also different approaches. So in terms of, for Margot, we're looking at an ASO that switches splicing and that doesn't, and not the RNase H degrading ones. And I think switching splicing might have less side effects than degrading the RNA. So that might be a little bit safer in that sense. But obviously that's why we're, while we're really excited that NLORM is developing this, we're also excited to be doing all these additional experiments in our mouse model, in our cells, to really, really deeply understand, the biology of these changes and the effects. And so I think, we're, want to make sure we take our time with really understanding what we're doing and having, that extra knowledge. But at the end of the day, it's always a gamble. I mean, it's always, you're taking a chance on a new treatment. And I think, we want to do as much as we can for the science to push it forward for the next kids and for the next, for the future, to try and gain as much as we can while being safe, but still, we have to try these things, because if we don't try, then we won't learn. So, I think, obviously we're not at a point where we have something we're ready to inject. And so I hope that we'll be able to collect enough data and enough, data that when that time comes, that we'll feel comfortable doing that, and based off of, the other patients. But yeah, obviously it will be scary for sure. But I feel like we have good team of people, and, people that we can talk to, and that we can, really get to a point where we feel comfortable. But I think we want to try and move things forward, because, that's the solution that we need, because drugs can only go so far. With these ASOs, we can try and fix what's happening, and not just kind of, use drugs to stop the seizures. Drugs have so many side effects. Just recently, now we're dealing with a side effect from one drug, and we can't go up on it because, it's causing these other side effects, even though it works well. And so that's really hard too. We've been through 12 different drugs, and, we need more, we need better treatments. And, this is a way forward, and we want to do what we can to make that happen.
Wow. Well, it just sounds like you have so many great resources and such a great network. I mean, and Lorem, as you said, a great resource for rare disease families out there. They're doing amazing work. Ionis, of course, like groundbreaking science, and they're just um, so laser focused on delivering these ASOs for a number of different diseases. If you go on their website, their pipeline is so impressive, and their time to clinic is really crazy, which what that means is they have a ton of really dedicated people just working around the clock on getting these things to patients, and it's incredible. Yeah, I'd love, as we wrap up here, if you have any insights you want to share for other mom scientists, other patient advocates, other people coming up in their careers. I mean, you have so much, that you've shared already, but just any kind of shout outs or places people can find you.
Yeah, I think anywhere, what's really helped me is finding my community, finding people that are going through similar things, similar processes, similar experiences, to share, to make you feel like you're not alone, to help you with your struggles, and celebrate your successes. So whether that's, as a postdoc, finding people that I love to work with, and who were really fun, scientists that I got to learn from and do cool science with, and move on into our careers. As a junior PI starting my lab, we have this new PI Slack group, which I highly recommend. All these junior faculty sharing their ups and downs and their successes. Even if you're not in person at the same place, interacting and learning from others. And similarly, from the rare disease side, I've really embraced this journey that we're on, and I've connected with so many rare moms and medical moms, and both here locally where we live, and also on Instagram and in Facebook groups, and just being able to talk to people who are going through what you're going through is everything. And it makes you just feel less alone, and gives you the comfort, and then the confidence that you can tackle these different things. So I think finding that community for me wherever I've been has been so important in helping me with the challenges and succeed.
That's awesome. Yeah, we're all about finding your community on Lady Scientist podcast. I love hearing that there's a Slack channel for new PIs. That's amazing. I mean, it's so important to reach out to your network and lean on others who are going through what you're going through. And I think you said something at one point in the conversation about realizing that you could do it, like connecting with this other PI, this other professor, and him allowing you to realize that you could shift focus and study epilepsy in your lab. And I think those are the moments that like I want to give to other people, you know, through this show, and allow them to just see, oh, I could do that. Like I could go after that. I could, you know, pursue a career in academia, or I could study this, you know, disease that we don't have all the answers for yet. So I really appreciate you taking the time to share your career journey and your experience as a mom, and as your experience as a patient advocate for your daughter. And I'm so hopeful, like, for children out there with the SCN8 mutation. Like, so many great people are working on this, and um, I I just feel very hopeful that, like, you know, however long it takes down the road, there will be a treatment course and an answer for these patients. So thank you for all of, like, the work that you've done, and the work you continue to do in this space.
Thanks for having me.
Yeah, thanks so much for being on the show. That wraps up my interview with Dr. Madeleine Oudin. Madeleine, thank you so much for taking the time and sharing all of your insights with us. I learned so much. If you enjoyed this episode of Lady Scientist Podcast, please give us a subscribe, leave a comment wherever you listen to podcasts, and I recommend you check out some of our other interviews. I interview several female founders of different biotech companies, and I'll be doing some more interviews with other mom scientists like myself. I am excited to bring more of you on this path of learning from other moms and scientists out there. So thank you again for checking out Lady Scientist Podcast, and we'll see you next time.